It's a pleasure to be here today to celebrate the accomplishments of this center and the department in which it's housed, and also the people of whom it's made up. I could go on at length about that and the influence that this place has had on my career working in a U.S. university, but they've only given me 20 minutes, so I'll have to save that for the reception and get on with my talk. It's interesting that this is a Center for Energy and Environment, Indoor Environment and Energy, because my talk is about a technology that's often applied with indoor environmental quality in mind, but I'll give a little summary of some research that we've done recently on the use of the same technology to save energy. And this is ultraviolet germicidal irradiation. This is also an interesting topic, I think, to bring up here because the focus in this center, from my perception, has been very much on ventilation and on the prevention of exposures and on human response. In the U.S., I think perhaps we've paid more attention or given more credibility to the possibility of using air cleaners, and I think this is one of the technologies going forward that's going to help us maintain high standards of indoor environmental quality under changing outdoor environmental conditions. Ultraviolet germicidal irradiation is specifically a technology for controlling microbial contaminants, indoors, outdoors. They can be in the air and they can be in surfaces. I don't think it takes too much to motivate the importance of this topic. Viruses, bacteria, fungi, they can cause infectious diseases, they can cause allergies, asthma attacks. Mold is a big problem in buildings, particularly in climates that are high humidity as this one is. And we also have the issue of biofouling of various components in buildings, plumbing systems, but also cooling coils in HVAC systems. The control methods we have available for air are dilution with ventilation, filtration, capturing biological particles out of the air, and the third mode, which UVGI falls into, is inactivation, inhibiting or preventing the reproduction of viable microorganisms. On surfaces, one of the most important methods is moisture management in the building. So if we can keep a building dry, then we're not going to have mold growth. Barring that, chemical cleaning and mechanical cleaning are options, and again, inactivation of microorganisms on surfaces is another possibility. These different approaches have different levels of effectiveness in different environments. They cost different amounts, and they may have secondary effects, some of which are adverse, that differ from one to the other. For those who are not highly familiar with this technology, I'll give a quick overview of how it works. It's been known for well over 100 years that certain wavelengths of light will affect the growth of microorganisms, specifically UVC and UVB radiation. What they do is damage the DNA or RNA of microorganisms. If you think back to your biology in DNA, we have 4 bases: adenine, thymine, cytosine, and guanine. Adenine and thymine go together. Germicidal radiation can break the adenine-thymine bond. Thymine dimers form. If you form enough of those, you make it impossible for the microorganism to reproduce, and it might as well be dead, although we don't use killed as the description of that. So the germicidal effectiveness of different wavelengths has been studied. You can see in the lower figure here the standard germicidal effectiveness curves. Most of the wavelength band that's highly effective is UVC, and 265 nanometers is about the most effective wavelength. This has been capitalized on in technologies for water treatment, for air and surface treatment. The sources that we currently use to generate germicidal irradiation are frankly no different than fluorescent lamp technology with a few enhancements. We use low-pressure mercury vapor lamps. Quartz tubes are used in these lamps so that the germicidal wavelengths will be transmitted. They produce mainly 254 nanometer radiation, which is about 80% of the maximum germicidal effectiveness. About 20 to 30% of the power input to a lamp goes into producing germicidal radiation. The rest is dissipated in a few other weak spikes at different wavelengths and as heat. One of the big concerns, aside from the lamps containing mercury, about this technology is the production of ozone. Wavelengths below about 240 will produce ozone, and there is a spike at 185 that's produced by a low-pressure mercury vapor lamp. The figure in the lower right corner here shows the transmittance of different types of quartz glass, and you notice that the green titanium-doped quartz admits almost all of the 254 and filters almost all of the 185. So there are some very low ozone-producing lamps, but if you misapply and use one that has the synthetic quartz, then the ozone-producing wavelengths will be emitted as well, and ozone may be a problem. UVGI disinfection follows standard disinfection theory to a first approximation, the Chick-Watson equation. So there's an exponential relationship between the dose that's delivered to a microorganism and the survival of a population. So the dose in this case is the fluence or the intensity of the radiation multiplied by time and modified by a microbial response coefficient K, which varies widely by orders of magnitude from one microorganism to another. So in the figure here, we see an example with 500 microwatt per centimeter squared radiation intensity over time varying from 0 to 2 seconds, and anthrax, which is very resistant, is hardly affected at all in that time period, but Legionella is reduced quite substantially. So one needs to know not only the characteristics of the lamps and the system, you need to know what organism you're trying to control and to what level when applying it. UVC is not good for microorganisms, so it's probably not surprising to find that other carbon-based life forms like human beings also have an adverse response to it. It's not one that is lethal or long-term, but if you are exposed to a lot of UVC radiation, you will get skin irritation, painful eye irritation, and the figure here shows the NIOSH, the U.S. National Institute of Occupational Safety and Health standards for UVC exposure. So if you're going to design a system that's going to be in an occupied space and people might be there for hours, you can see you need to have a very low intensity, something approaching 0.1 microwatts per centimeter squared, to be able to do that safely. Systems that are put into HVAC for disinfection of air over the course of a second or two may have hundreds or even thousands of microwatt per centimeter squared, and you can see from the allowable exposure time there that in that case you simply can't be exposed to those lamps when they're on unless you're fully clothed with protective clothing. So there are some safety issues here in applying, but that happens with other technologies as well. So those are the basics. The main types of equipment that we see in buildings are, first of all, in-room systems described as upper air systems, where there are fixtures that are placed above the occupied zone in the room. You can see two of them glowing blue in the figure over on the far left. And those create a fairly intense zone of irradiated air above the occupied zone, into which air from the occupied zone moves either by natural convection or under the influence of the ventilation system. That's illustrated in the middle figure. And this has been applied for a long time with a fair amount of evidence in the field in the laboratory that it works. One small example here is the Harvard School of Public Health experiment from the 1970s, Riley and Middlebrook, where they estimated that the effect of upper air UVGI could be the equivalent of 10 air changes of ventilation in some circumstances. That's illustrated by the differences between UV and non-UV reduction of microbial population in the figure there on the right. Other types of in-room systems include self-contained air cleaners that might have lamps inside of them. These have all of the same operating issues of any other portable in-room air cleaner. One of the big issues is how well do they turn over the air, but they can be quite effective for single pass effectiveness. On the right is a unit that's intended for unoccupied use to disinfect contaminated facilities like operating rooms or holding tanks at jails or other facilities that may be highly contaminated and where there's a period of time where they can be disinfected when no one is present. In HVAC systems, the most common way to apply UVGI is in an air handling unit, although we refer to those typically as in-duct systems. Sometimes they are simply put into ducts in the system. But the reason for putting them into an air handling unit is twofold. One is we have much lower velocities inside the air handling unit than we typically have in ductwork, so exposure time can generally be longer. And the other reason is that we can have a system that disinfects air and at the same time, if it's positioned in the right place, can irradiate a cooling coil and control biological growth on the coil and also in the condensate pan. There's some interesting application issues with these systems that are illustrated by the figures over on the right-hand side. At the top, we have what's called the wind chill curve for a low-pressure mercury vapor lamp. The plasma inside the lamp has its output controlled by the dew point and by the lowest temperature on the lamp surface. At about 40 C, you get maximum output, and if the environment results in the lamp surface being either warmer or cooler, the output decreases, and that leads to maps of performance like the one that we produced from some of our experimental data shown below, where you can see that over a range of temperatures and velocities, we may have 95% maximum to 35% minimum in conditions that may actually occur in a system. So that's something that has to be considered in design. The other type of system I'd like to highlight here, because this is the research I'm going to mention at the end, is UVGI for cooling coil maintenance. I already indicated that these systems are sometimes installed where they can irradiate the cooling coil. When you have a coil that's doing dehumidification, that's wet much of the time, it will collect microorganisms and slime will grow on it, bacteria, fungi, and that will reduce the space between fins, increase pressure drop, and it also acts as thermal insulation and will decrease the heat transfer coefficient. So if we can control that, we should be able to get some improvement in energy performance, maintain intended airflows, and perhaps also prolong the life cycle of the coil. So UVGI can do that, and it's an alternative to some not highly satisfactory alternatives like power washing, which often tends to simply push debris into the center of the coil where it's more inaccessible and packs it together than actually removing it. We can see someone doing that. Sometimes chemicals are used, which themselves create indoor environmental quality problems. And then the third approach to coil maintenance is neglect, which is also quite common. So many of them just become more and more fouled over time. So this can be done either as the main purpose, which generally requires less power because you're irradiating a stationary object, or it can be done coincidentally to air disinfection, which requires a larger system. A little bit of the very interesting history of UVGI. Some of the fundamental physics was research in chemistry in the, actually in the 19th century. There was already a working water treatment plant using UV in Marseille, France in 1909. Upper air applications were first made in the 1930s. HVAC in-duct type of applications in the 40s, and the growth control systems started to emerge in the 70s. The World Health Organization approved UVGI for tuberculosis control in the late 1990s, and in 2005, with growing interest, ASHRAE formed a technical committee on the subject. The last time I looked at where the market was heading, it seems to be growing at about 10% per year. It started very small, but it's growing very rapidly in the HVAC sector. I could go through a number of papers to try to convince you that there's evidence that UVGI is actually effective. I'll only highlight two here that are interesting to me, particularly because they're field studies. This first one was done in 1942, published in 42, by Wells and Wilder, and from Harvard School of Public Health. This looked at incidence of measles in schools in the Philadelphia area, and they installed upper room systems in some classrooms and not in others, and tracked for a fairly long period of time the difference in incidence of measles. And you can see in the figure on the right-hand side, the top and bottom are to the same scale, that there was about a 3 to 5 times difference between the incidence of measles in the classrooms that had no UVGI, and the ones that had an upper air system. So that's been a fairly durable and persuasive result over a long period of time. A much more recent study was done by Menzies, Canadian, and published in The Lancet. This was looking at sick building syndrome symptom incidence in 2 office buildings where UVGI was installed in the air handling units, and the study was run double-blind. So neither the researchers nor the occupants knew when UVGI was operating. They surveyed for sick building syndrome symptoms during both periods and also did measurements in those buildings of the surface and air microbial and endotoxin concentrations, and found large reduction in concentrations on surfaces and air, and concluded there was a statistically significant reduction in the SBS symptoms. This was, again, a very persuasive study, but there haven't been enough yet. That's true of many topics in our field. At Penn State, we have worked mainly on in-duct type of systems. We've looked at modeling of dose, at measurement and modeling of lamp characteristics, at the standardization of K values, which are not very well known for many microorganisms of interest. And we've done some work on the economics as well. The most recent study we've done was on cooling coil irradiation, partly with funding from ASHRAE, and 2 studies in the US, sites in the US were studied by one of my students under an ASHRAE research project, and we collaborated with some folks at the National University of Singapore on 2 systems there. So we wanted to answer questions like, does coil irradiation actually reduce pressure drop and increase heat transfer coefficient, and how much? Surprisingly, after decades of application, no one had ever done a peer-reviewed, repeatable study to answer those questions. Where does the energy use change and by how much? And are there climate-related variations in the effects? Is it economical? And since we're irradiating the air as well as the coil surface, what would the potential value of the air disinfection be as well? These were all questions that had not been looked at until about 5 years ago. So we did, as I already mentioned, field studies in 2 locations. In the US, we had a fairly temperate location at State College, where my university is. That coil was dry for much of the year. The other was in Tampa, which is a fairly hot, humid climate, although not strictly speaking tropical, and then systems in Singapore where it's hot and humid all year. So we installed commercial UVGI systems and instrumentation to do energy-related measurements, and we monitored for a period of months to develop a baseline, then turned the systems on and obtained post-UV data and analyzed it to determine all the things that I've already discussed, changes in delta P and U. And then we fed that into energy modeling that was done parametrically for different locations and building types. And we also did the collateral IAQ benefit analysis. So here are some photos of what these coils look like before you apply UV to them. We didn't ask someone to neglect their coil. We went out and looked around in mechanical rooms and found coils that looked like this one at the top that had mold growing on it that was sticking out perhaps a centimeter. So we had a very fouled system in Tampa to work with. In Singapore, they looked for more normally fouled systems, but still those coils looked like the ones at the bottom of this figure. This is an illustration of the instrumentation package we put in. Nothing particularly exciting here. Temperature and pressure difference and relative humidity and flow sensors. On the right, you can see running across the middle of the air handling unit face the UV lamps that were installed. So here's what happened with the Tampa system from before UV to after. These are photos taken in more or less the same place, and you can see 2 things here. One is that the mold does not look nearly as happy to be there as it was before we started irradiating it. And another thing that happens in most of these coils, and I'm not sure if it actually means that they're cleaner or not, is that the metal surfaces seem to look shinier after irradiation for a period of time than before. And this all happens over about a period of a month. We found that once we'd gone through a month of continuous irradiation that the conditions were pretty stable. Pressure drop results for one of the Singapore buildings are shown here. The red data illustrate the pressure drop versus flow before UVGI was operating, and the blue after, and there was a quite good correlation in the change before and after, about a 15% difference in pressure drop. That was good news for us because we found something significant. The bad news was that the industry had been claiming much better performance, which is something that happens all the time, but this is why we do this work. Heat transfer, similar magnitude of results. The heat transfer coefficient, either overall or air side, went up fairly significantly. On the air side, nearly 15% in the maximum case, and we found that translated into a measurable decrease in chilled water flow rate and increase in chilled water delta T, which reduced pumping energy, and there were also some benefits to the chilled water plant itself. So if we take those results and embed them in an energy simulation of a whole building for a year, what we found was for an office building in Singapore, about a 9% reduction in fan energy, 6% reduction in pump energy, and a 0.4% reduction in chiller energy. But interestingly, because the compressor on the chiller consumes a lot of power, 48% of the energy savings were from that 0.4% impact on the chiller, at least in the simulation. I don't have time to cover the parametric study that we did on US locations, but what we found was that in the best cases, this was the sort of result we would get, and in the worst cases, it would be much less significant because of the climate. If you don't have a lot of continuous growth of biofilm, then you don't have a problem. The results, very briefly, from modeling the indoor air quality impact, we did what others have done in the past, which was to take the Wells-Rayleigh equation and to use it to estimate the impact of reduced exposure to infectious microorganisms on sick leave. We looked at different, and other metrics as well. We looked at a number of different types of occupancies, so hospital-acquired infections and dollies were also looked at. We found, as these studies often do, that if you monetize those benefits, they're larger than either the energy savings or the maintenance cost savings of using UVGI, but they're subject to credibility issues. Interesting work that I don't have time to cover in full detail, but it's in an ASHRAE research report and in several papers we've been publishing over the last year and over the coming year. So to conclude, there certainly is a need for indoor microbial control, and one of our options for doing that is to use UVGI. There are a number of knowledge gaps that still need to be filled and could be filled by researchers here and other places. Clinical effectiveness instead of simply what are the reduced exposures, better understanding of performance characteristics for design, the development of mercury-free zero ozone production sources like LEDs, which haven't been commercialized yet. And I think also there's not enough work done on secondary air pollutant generation. All those things remain for the future. And so with that, I will conclude. Thank you.